Chapter 10 is on the cardiovascular system, and this includes the blood and the heart. Now, this recording that you're going to have here in this video is just the blood, and then the heart sections of the lecture are already in Canvas, and they're broken down into different segments. So I'll stop after we discuss the blood portion of the chapter. So we need to understand that blood is basically a type of a connective tissue which is found in the human body in a fluid state. It is made up of plasma, which is highly viscous liquid, and 3 different types of blood cells are floating around in it. So blood is a combination of plasma, which is a watery liquid, and cells that float in it. And it supplies essential substances and nutrients such as our sugar, oxygen, and hormones to our cells, and carries waste away from these cells. And this waste is eventually flushed out of the body in urine, feces, sweat, and then of course our lungs being the CO2. Some functions of the blood are transport substances such as oxygen, nutrients, waste products, and hormones. So blood is the primary means of transport in the body that transports important nutrients and materials to and from the cells that make up our body. The first duty is to take the oxygen from the lungs to all the cells of the body, and then to collect the CO2 from the cells and deliver it to the lungs. That's your carbon dioxide. It also collects metabolic waste and takes it to the kidneys for excretion. So blood delivers nutrients and glucose from organs of the digestive system to other parts, including the liver. It can regulate your pH. It can help regulate pH, prevent excessive blood loss by hemorrhage, and of course fight infection, being your white blood cells. 2 components of blood. We have the living blood cells, which is the formed elements. 45% are your red blood cells, white blood cells, and platelets. And then secondly, the 55% is your plasma, which is your non-living fluid matrix. And blood can be separated into 3 layers. You have the red blood cells, which consist of the hematocrit portion, the middle, which is the leukocytes, which is your white blood cells and your platelets, and then the plasma is the top portion. So this photo just depicts that. So if you were to have your blood drawn in a lab and they centrifuged it, it would separate, and that is how it separates itself. Physical characteristics of blood: if it is oxygen rich, it has a scarlet red color. Oxygen poor is more of a dull red. Its pH does range from 7.35 to 7.45, and of course the volume can average just a slight bit between males and females. So blood cells are formed in the bone marrow, and all blood cells arise from the same bone marrow stem cells. And stem cells are immortal. That means they never die until you do. So stem cells are pluripotent, meaning they have the potential to become any type of blood cell, which gives rise to erythrocytes, which is your red blood cells, leukocytes, which is your white blood cells, and your platelets. So the formed elements making up the 45% of the component, remember the other 55% is the plasma, which is the non-living matrix. But the formed elements here making up 45%, we have the erythrocytes, that's known as your red blood cells, and those function specifically in oxygen transport. Your leukocytes, which is your white blood cells, specifically function with your immunity. And your platelets and your thrombocytes, or your cell fragments, this functions in your blood clotting. So getting into each blood cell individually, let's talk about the erythrocytes, which is your red blood cell. They contain hemoglobin. They are red in color due to the hemoglobin, and they are flexible and change shape when flowing through our capillaries. So as blood travels through the lungs, molecules containing oxygen attach themselves to the hemoglobin, and the hemoglobin then releases the attached oxygen to cells as the blood travels through the body tissues. And the now empty hemoglobin will bond with tissues that contain waste gases and carbon dioxide, transporting them away from the body. So the main function, of course, is to carry oxygen, and it is the most abundant formed element. And here is some characteristics. It has biconcave disc. Essentially, it is a bag of hemoglobin. It has no nucleus, and it contains very few organelles. So it does not have a nucleus. It does not have a mitochondria or ribosomes. So because this red blood cell does not have a nucleus, it cannot repair itself. Now that's important to understand. So let me explain that. If you recall from chapter 3, talking about and learning about mitosis, replication is the division of the nucleus, which is mitosis. So it is the division of the nucleus. So you have to have a nucleus which contains your DNA information to be able to copy it and make another copy of it, another form of it. That's how in mitosis we get 2 daughter cells out of 1, and they're identical. So because a red blood cell does not have a nucleus, you need to understand that they cannot repair themselves. They cannot replicate and divide. They are made in the bone marrow, so they live about 120 days approximately. And remember, they can't repair. So when they get worn out, they die, and the body is going to replace them. They are destroyed and recycled by the liver. So without red blood cells, the body would slowly begin to shut down. They contain our hemoglobin, which of course is that protein that contains the iron, and again is responsible for transporting our oxygen into our cells, or to our cells, and then the carbon dioxide out of the body. So we have to have our red blood cells in adequate supply in order to supply our body with oxygen. So with erythrocytes, again, the hemoglobin is the iron-containing protein that transports oxygen from the lungs to our body tissues and cells. 97% of a human's red blood cells' dry content is protein. It does bind strongly but reversibly to oxygen. So we have oxyhemoglobin, which is the hemoglobin that is bound to the oxygen, so it's bright red in color. Then we have deoxyhemoglobin, not carrying oxygen, so it's dark red in color. So because they cannot repair themselves when damaged, they have a very short lifespan of about 120 days, as I mentioned. The removal of old and dying red blood cells is carried out by the spleen, and 2 to 3 million red blood cells die every second. So the rate of red blood cell production must equal the rate of red blood cell death. And red blood cell production occurs through a process called erythropoiesis. And we talked about hematopoiesis just a little bit in the cell chapter in chapter 3, but here we get into it a little bit more distinctly on the next slide. So our red blood cell production, also known as our erythrocyte production, is dependent on oxygen concentration. Homeostasis is maintained by negative feedback. Remember, most of your examples are going to be negative feedback, getting you back to normal or homeostasis as soon as possible. And you need to understand here that if blood oxygen concentrations are low, and this is again related to the negative feedback system. So if blood oxygen concentrations in the body are low, that's known as hypoxia. So the kidneys and the liver are going to be stimulated to release a hormone called erythropoietin. And that hormone is going to stimulate the process called hematopoiesis in the red bone marrow. So when the red blood cells increase, so does your oxygen level. So increased oxygen levels will decrease erythropoietin. And remember, that's the whole point of negative feedback, is shutting off the stimulus when we reach the point of homeostasis. So remember, the process of making more blood cells is hematopoiesis that occurs in the red bone marrow. So when that happens and we get more red blood cells, the more red blood cells we get in our body, the higher our oxygen content is. Because remember, we have the hemoglobin, the iron-containing protein that our oxygen attaches to. So you need to understand what that process is all about. So hematopoiesis is the production of the formed elements of the blood. So the erythropoietin is that hormone produced in the kidney and through negative feedback systems in which tissue oxygenation controls erythropoietin production. It provides homeostasis and oxygen delivery to the body tissues. So as the oxygen carrying capacity of red blood cells decreases due to high altitudes, for example, the body's requirements for O2, or oxygen, is going to increase. Thus, new red blood cells need to be produced, and the drop in oxygen carrying capacity is referred to as the hypoxia, which in turn is detected by cells within the kidney. So when low oxygen levels are detected, these cells within the kidney release that hormone called erythropoietin. That's going to stimulate the red bone marrow to produce more erythrocytes, or red blood cells, through the process called erythropoiesis or hematopoiesis. So this causes that increase in red blood cell count and thus an increase in oxygen carrying capacity of the body. This increase is again detected by the cells in the kidneys, and the release of erythropoietin is then decreased because we don't need it anymore, restoring the oxygen carrying capacity back to normal. So factors necessary for the hematopoiesis process is iron for our hemoglobin and folic acid and B12, which is necessary for DNA synthesis in the red bone marrow. Hemocytoblasts, these are stem cells in the red bone marrow that produce blood cells, all of them. You get your red blood cells, your white blood cells, and your platelets. So erythrocytes, again, I've mentioned, live around 120 days. They are destroyed and produced daily, and worn out red blood cells are removed from circulation in the liver and the spleen by macrophages through phagocytosis. You learned all of those terms back in chapter 3 in the cellular chapters. If you need to go back and review that content, please do. So everything in this course builds on one another. So please make sure that if you are unfamiliar with some of the terms, because you need to know what macrophages are as well as phagocytosis. So make sure that you go back to chapter 3 and review that. Contents of red blood cells are recycled to produce more. So when a hemocytoblast divides into two daughter cells, one of the new cells becomes a blood cell. The other remains a hemocytoblast, ensuring that there's always enough hemocytoblast available to continue to arise more blood cells. Next, we get into the leukocytes. The leukocytes are our white blood cells. These are crucial in the body's defense against disease. They do not initiate the clotting process. The white blood cells is all about defense mechanisms. Red blood cells is all about oxygen carrying capacity. So these are complete cells. They do have a nucleus and organelles. It's a little different there from the red blood cells who did not have a nucleus and very few organelles. They are able to move into and out of blood vessels. That's known as diapedesis. They can move by amoebid motion. They change shape to squeeze through the tissues. They can respond to chemicals released by damaged tissues. That's known as chemotaxis. And we need to understand that they are colorless cells that possess a nucleus. They act in defending the body against pathogens. White blood cells are the cells of our immune system, so they defend the body against infections and foreign material. White blood cells will also fight abnormal cells, such as cancer cells. So this chart here just goes over and shows you kind of a different, it shows you the description, function, that type of thing. Don't pay attention to the number. I definitely don't want you to memorize that. But you do need to know your formed elements there, and of course, your red blood cells, your white blood cells, and then your platelets. But most importantly, pay attention here on this chart. It divides your white blood cells for you, which you do have to know your granulocytes versus your agranulocytes. So your granulocytes are your neutrophils, your eosinophils, and your basophils. And then your agranulocytes are your lymphocytes and your monocytes. And all of those are a type of leukocyte, a white blood cell. So the description and the function is what is important here on this table. So we get into, these are just a few pictures here of our white blood cells. So the first one that we're going to talk about is your neutrophil. And the neutrophils are the most numerous. They are an active phagocyte. They're the first to arrive at infection sites of damaged tissues, and they are the most abundant during acute infections. Next, we have the eosinophil. These kill parasitic worms, and they are found in response to allergies to lessen inflammation. The next one we have is our basophil. They are the rarest of the white blood cells. They release histamines, which stimulate inflammation, and heparin, which inhibits blood clotting. And then we have something called a mast cell. This is a basophil that have moved into damaged tissues to start inflammation. Then we have a lymphocyte, found mostly in lymphatic tissue, plays a major role in the immune response, and B cells produce antibodies, and T cells attack directly. A monocyte. This is the largest of the white blood cells. They function as macrophages in tissues, and they are found in large numbers in chronic infections. Then we move on to our platelets, also known as thrombocytes. This major function is in the clotting process to slow blood loss, derived from ruptured multinucleate cells called megakaryocytes. They clump together to plug breaks in the blood vessel. So hemostasis. Hemostasis is the stoppage of blood flow. So this is a series of reactions that stop blood flow, like maybe following a major cut of some kind. So it involves 3 stages here. We need to understand that damage to small blood vessels and capillaries frequently occurs. So when these vessels are damaged, there are 3 basic mechanisms that promote hemostasis, or the stoppage of bleeding. Following damage, there is an immediate reflex that promotes vasoconstriction, thus diminishing blood loss. So number 1, vascular spasm. So vasoconstriction, which decreases blood loss. Vessels constrict in response to damaged tissues, and the platelets release serotonin, a hormone that causes vasoconstriction. Secondly, we have platelet plug formation. The collagen fibers are exposed and the platelets stick to the fibers, forming a plug. And then last, we have coagulation, and that's your actual blood clotting. So blood clotting begins 30 seconds to several minutes after phases 1 and 2 here have finished. The process involves the formation of the insoluble protein fibrin that forms a network of fibers which traps blood cells and platelets from forming a clot. You need to understand there that serotonin, I have it underlined there, the hormone, this is the chemical that is released to bring about vasoconstriction during the vascular spasm phase. And sudden and severe loss of blood can lead to shock and death. So when blood vessels are damaged, this hemostasis, also known as our clot formation, will arrest or stop the bleeding. So here, clot formation is a complex process, but it is completed within 3 to 6 minutes after a blood vessel has been damaged. The clot is restricted to the site of damage because that is where proagulants outnumber the anticoagulants. So hemostasis, the steps in coagulation here. Step 1, the injured tissue or the vessel releases thromboplastin, and collected platelets release platelet factors. Both thromboplastin and platelet factors react with clotting factors in the plasma to produce prothrombin activator. Step 2, in the presence of calcium, prothrombin activator converts prothrombin, which is an inactive enzyme, to thrombin, an active enzyme. Step 3, in the presence of calcium, the thrombin converts fibrinogen, which is a plasma protein, into hair-like insoluble fibrin. This forms the meshwork that traps red blood cells and forms the basis of the clot. So for this reason, blood clots are sometimes called fibrin clots. So because this compound forms the meshwork of a blood clot, the clot is often called a fibrin clot. So after a clot has formed, the fibrin strands contract, which produces a more compact clot and pulls the damaged tissues closer to each other. So hemostasis consists of 3 processes: blood vessel spasm, platelet plug formation, and blood clot formation. And this is just a photo of a blood clot where it shows blood cells trapped in a meshwork of fibrin strands. So when a clot forms, it pulls the damaged tissues closer together. Blood plasma, this is composed of approximately 90% water, and patients all over the world rely on plasma therapies to treat rare chronic diseases. Plasma is referred to as the gift of life because it is the essential starting material for these therapies. To donate your blood, you have your blood drawn. Well, plasma will be separated from your blood, and red blood cells return to your body through plasmapheresis. So first plasma donation will take about 2 hours, and the other one's only about 90 minutes. And plasma is used to treat bleeding disorders where the patient is unable to clot. They can treat immune deficiency disorders who do not respond to antibiotics, treat lung diseases, liver diseases, treat burn victims by providing albumin, treat rabies, tetanus, dialysis patients, organ transplant recipients, pregnant women with Rh incompatibility. So plasma is very, very, very important. So it includes many dissolved substances, nutrients, salts, respiratory gases, hormones, proteins, and waste. Some of the plasma proteins, albumin forms 60% of plasma proteins, regulates osmotic pressure, keeps water in the blood, maintaining blood volume and pressure, serves as a buffer to regulate pH. Globulins, alpha and beta, carry lipids and vitamins. Gamma consists of antibodies. Fibrinogen plays a vital role in blood clotting when it's converted to fibrin by thrombin. And blood types are determined by the presence of specific antigens on the plasma membranes of the erythrocytes. So if you have antigen A, your blood type A, antigen B, blood type B, and so forth. So notice that group O can be given to people of any blood type because it's the universal donor. And then patients with the group A can only accept type A or type O blood, and patients with type AB can receive from any group because they're the universal recipient. So if a different antigen is introduced into the body, they will be attacked by antibodies present in the plasma. This will cause the foreign red blood cells to clump together, called agglutination. So the blood type determines what antibodies are located within the blood. An individual possesses antibodies against the antigens they lack. So for example, type A blood has type B antibodies, so they're anti-B. And if type B is put into the body, the immune system reacts as if it were a foreign invader. So the antibodies will clump the incompatible blood, and that's called agglutination. And this photo just depicts that antigen, antibody, and how the blood type is determined. And here, the universal donor, as I mentioned, is O negative. It has no antigens on the surface of the erythrocyte. Therefore, it can donate to anyone because it will not trigger an immune response. The universal recipient is AB positive. It has all antigens present, so it can receive any type of blood. So here, just as there are major blood groups such as type A and type B, there also is an Rh factor. The Rh factor is a protein that can be present on the surface of a red blood cell. Most people have the Rh factor, so they are Rh positive. Others do not have the Rh factor, so they are Rh negative. The Rh factor is inherited, so if the mother is Rh negative and the father is Rh positive, then the fetus can be either. The Rh factor can cause problems if you are Rh negative and your fetus is Rh positive. These problems usually do not occur in the first pregnancy, but can occur in later pregnancies. So when an Rh negative mother's blood comes into contact with the blood from her Rh positive fetus, it causes the Rh negative mother to make antibodies against the Rh factor. These antibodies attack the Rh factor as if it were a harmful substance. So during pregnancy, the woman and the fetus do not share the same blood systems. However, a small amount of blood from the fetus can cross the placenta into the woman's system during birth. So if you are Rh negative, you are given a RhoGAM shot, that's what it's called, and this prevents the production of Rh antibodies. And it's given to an Rh negative mom around 28 weeks within 72 hours after delivery or even after a miscarriage. So the dangers, again, it's all due to the immune system being sensitized after that first pregnancy. And this means that the mother will begin to produce more of those antibodies to fight it off. So in a second pregnancy, the antibodies in the mother's immune system could attack the Rh positive blood and lead to something called erythroblastosis fetalis, hemolytic disease of the newborn, where the baby would be anemic, hypoxic, and cyanotic. So the skin would be blue from lack of oxygen. Brain damage or death can occur unless transfusions are done to provide more red blood cells for oxygen transport. So the erythrocyte disorders, I'm not going to read all these for you, but you just need to know what these are. But you have anemia. So you have different types of anemia here: nutritional anemia, hemorrhagic anemia, pernicious, hemolytic, aplastic, and sickle cell. And then erythroblastosis fetalis and polycythemia. And some leukocyte disorders are infectious mononucleosis, leukemia, leukocytosis, leukopenia. And then clotting disorders are hemophilia, thrombocytopenia, thrombosis, and embolism. So as I mentioned, this next section of the chapter is dealing with the heart in the cardiovascular system. So I just discussed the blood and the heart portion. This is explained in the individual recording clips that are broken down into different segments because they're rather lengthy, and so they're all broken down into different segments explaining the cardiovascular system pertaining to the heart.